Hello and welcome to the second anticipatory briefing. This time we're going to talk about the promises of synthetic biology. So in these sessions, we basically showcase the GESDA breakthrough radar technologies, and we tell you about what to expect from them for the next 5, 10, and 25 years. Our next speaker is a professor at Agricultural Genomes Institute in Shenzhen, Chinese Academy of Agricultural Sciences. It's very hard to say for me somehow. He's got a bachelor degree from the Department of Intensive Instruction in Nanjing University. Maybe it's a place where you get your degree in less time because it's intensive instruction. He got a master degree from the Tsinghua University before moving to the US, Iowa State, and Johns Hopkins University School of Medicine. Then he moved back to Shenzhen Institute of Advanced Technology, where he joined the Agricultural Genomes Institute. His research interests are many: the development of new technologies for gene synthesis and assembly, synthetic genomics, and also synthetic biology using different model organisms, and is one of the key members of the Synthetic Yeast Institute Consortium. He has published in many peer review journals, among them Cell, Nature, Science, Molecular Cell, Natural Communication, and Cell Genomics. He's going to tell us about synthetic biology, which goes beyond reading and editing genes by allowing us to write the whole genomes now. It's expected to have a very deep impact on fields like life science and sustainable agriculture over the next 5, 10, and 25 years. Please help me welcome Junbiao Dai.
Thank you. Thank Lauren for the nice introduction, and thank GESDA to give me a chance to talk about my work and also, what happened in the field and what's my view for the synthetic biology and especially synthetic genomics. So, as Lauren introduced, I was trained as a biologist, right? So, as a biologist, you will think about the cell is the fundamental structure of every living organism, right? So in the point of biologists, in the eye of biologists, you view cell as a dividing machine. They were replicating itself and dividing into two cells and keep dividing. And also, I'm also very interested in engineer. As an engineer, what you look at the cell, you were thinking about this is a micro manufacturer factory, right? The cell are actually making, while I'm speaking and you guys are listening, the cells are working in our body, and they were making all different stuff, right? Like the moleculars and replicates DNA and the replicating itself. They're making tons of material and stuff. So that's just like the operating systems are operating the computer systems. Similarly, in cells, there are also an operating system, which is the genome. Okay, so the genome, as the central, genetic information for each cell, they were stored in the nucleus for a eukaryotic cell, and for a bacterial, they were just stored within the cell. And nowadays, to understand the operation system, what we want to know is what is exactly this operating system was written, and what is the alphabet of this operating system. So when the technology developed, we were able to sequencing the genome of an organism, which leads to the digital code, as we can see here, which is A, T, G, and C, which compose as the entire central operating system. So one of the famous projects, which is trying to understand the human operating system, how the human can grow and can develop and can be healthy or get ill. One project was proposed a long time ago, back in 1980s, which is the Human Genome Project. It took about 15 years, and spend $3 billion US, and involves over 1,000 scientists from 20 institutes among 6 different countries to conquer this project. And the consequence of this project bring us is one of the technology breakthroughs. Before the human genome sequencing project, people are trying to sequence in the genome as a fragment like 500 to 1,000 base pair fragments using Sanger sequencing, which is also called first generation DNA sequencing. So during the human genome project, a lot of technology was developed. One of them is the high throughput technology, which also called a next generation or second generation technologies, which allows us to sequencing millions of fragments at the same time, simultaneously, to read all the DNA sequences. Therefore, it allows us to do sequencing the genome at a much larger scale and much faster pace. And now we also have the third generation sequencing technology, which is to sequencing the single molecular, and it's going to be tens of it can sequence tens of KB fragments on average. So that's also called long read sequencing. So with the newest technology, there's several giants in the world. As I demonstrated here, showed here, which is announced by the Chinese company called BGI. They announced the newest sequencing machine called the T7, the MI6 T7, which is able to sequencing 6 terabyte of DNA sequences within a day. That counts about 10,000 human genomes a year with a single machine. So you can imagine, right? For the first human genome sequencing, we spent 15 years. And now, in 1 year, we can sequence 10,000 people. The developer of the technology not only increase throughput, but also bring us a dramatic decrease on the price. If you look at back in 2001, which is announced the completion of the human genome project, the project sequencing 1 human genome cost about $1 million US. And now, with all this technology development, now you can sequencing your own genome with less than $1,000. So that is the huge job, way more than the exponential cost reduction we can see as a Moore's law, right? Because of the capacity increased really rapidly, and it also gets more demands. So this is a chart showing the global sequencing market, which is actually increased with a much higher annual rate, about 15%. So right now, if you look at in 2020, the market is about $10 billion US, but in 2032, it will reach $40 billion US, the entire sequencing market. So with that market, with that also demands, because the demands is coming from the technology development. And also, it allows us not only sequencing 1 human genome. Now we can sequencing 1,000 genome, the 10,000 genome, or even potentially in the future, everyone in the world will get sequenced. Okay. So one of the purpose to sequencing a lot of the different people is to try and find a lot of those variants. Because we can sequence a bunch of the healthy people and also people with some genetic diseases. By reading all the compared, the people between the healthy one and the one carrying the disease here, we were able to identify those genetic variants, which are the causative effect leading to the disease. Okay, so once you are able to find those causative effect of genetic disease, what you're going to do, right? So for medical application, what you want to do is trying to cure disease. So how? One way, actually, another technology, many of those guys, you guys are all familiar with, is the CRISPR system, which is also called, gene scissors or genetic for the gene editing, which allows you, for example, as shown here, if you find that there's a variance which caused the disease, you want to fix it, right? As you can see here, if you got ATC, you have to fix it to CGT back to normal, and you can using this tool to do it. Okay. And by the way, this tool is also identified when you sequence all the bacterial genomes. Those tools exist in our genome bank. So all we need to do is try to identify them and use them. So this is, genome editing. However, if you think about one more question, right? What if you could write a genome? That means you can reorganize all the genetic information. You can rewrite each line of your script for the operating system, and therefore it will allow you to repurpose or reprogram anything which are biological. You can write, for example, the microbiomes, right? You can rewrite the genome, make it grow faster, make it become more suitable for certain, production of certain natural product, for example, and they will be make much better, cell factory. You can also rewrite the genome of a plant, for example. It will not only increase the yield, but also improve the food quality and the nutrition, and potentially you'll get more neutral food and for your personal health. And of course, you can also write the pig genome, for example, which is a very good donor for the organs for human to utilize, right? And because if you can write the genome, you can eliminate all the viruses the pigs may carry, and you can also, humanize the immune system to make it more suitable for organ, rejections, things like that. So, if giving you the power, if you to rewrite a genome, you can do a lot of different things. So there's a couple things has been done in the field. I'll just give you a quick overview about that. Okay, one work was done by Jason King from the MRC Laboratory of Molecular Biology, and what he did is, we all know as a biologist, the natural uses 64 codons for the triplet to encode 20 amino acids. Okay, what Jason did is in his lab is to eliminate these codons from 64 to 61. There's 3 codons which was removed from the genome, and that what the consequence is? That means you can remove the transfer RNA which carries that, the amino acid using that codon, and then the host, the bacteria, will become too resistant to its virus as a phage, for example, for the bacteria. Because you can imagine the phage is still using the 60 codons. They require those 3 codons to carry the right amino acid to its right positions. Now you remove these codons from the host. Now when a phage comes, the protein will not be able to translate it as the virus wishes, right? Therefore, you basically build a wall to block the infection of the viruses. So another example is we participate in a project we call the synthetic yeast. So many of you may be very familiar with budding yeast, right? It's an organism we use to bake bread, and of course for beers, which is more important, right? So we, together with Patrick and also a few different labs across the world, and we form a consortium trying to redesign the yeast genome, get version 2 of the operating system for these microorganisms. So through that project, what we want to do is we want to develop tools and platforms which allows us to develop technologies for genome assembly, because the genome is much bigger than the one previously did. We can also optimize the organization of the genes in the genome to make it more suitable for the cells to grow and to cells to improve resistances, fitnesses under stresses. And of course, you can faster the evolutions. With all those platforms and tools developed, we can using the synthetic yeast for many applications. For example, to make it better, brewer, right? Make the beer more testable, and makes it more for more better, cell factory for medicines, for other fermentation things, and also make it better for resistances. And recently, we're also doing something for plants. Okay, I don't know if you guys read the scientific fiction book written by Liu Cihang, which is a famous Chinese writer about 3 bodies. Okay, so when the weather or the condition goes bad, what you can do? Dry the stuff, right? Then you can store it there for a long time. We found there's one species in plant which can do the same thing almost. Okay, this is a plant species called the moss, which is a desert species found in Xinjiang province, which is growing in the desert. Usually, they will be dried. However, once you pour a bottle of water onto it, in seconds, they will turn green and make photosynthesis immediately. So which is very amazing. So we were using this system to explore if we can redesign the genome and try to understand how this resistance come from and how can we using the system to make it even better, or transport the system to other crops to increase the crop yield and crop resistances, for example. Okay, so my time runs out? Okay. Okay, so I'll go very quickly for what I view in the next couple years, for the 5 years, short, long term. The first thing is our vision is, we still have a long way to go. Okay. Since 2010, we built the first synthetic organism by J. Craig Venter Institute, which is only 1 billion base pair for the genome size. And when Jason built the E. coli genome, which is about 4 billion base pair, and the yeast is about 4 million base pair, and the yeast is about 12 million base pairs. So we have a long way to go if you look at genome size, right? From chicken or even pig or even a human, which is 3,000 megabyte. So there's long technology have to be developed. A lot of technology has to be developed. So the good thing is we are seeing there are technology breakthroughs coming, right? And we'll see the maturation of high throughput DNA synthesis. For example, we are using a lot of the chip-based synthesis and also enzymatic synthesis, which is, there's many companies are able to provide cheaper DNA synthesis, DNA synthesis now. And we're going to expect it that in the next 5 years, we're probably going to see a 10,000-fold decrease in the price. And that means we can resynthesizing a human genome with only about $300,000 US. And with that ability, it allows us to increase the productivity of the DNA synthesis and initiate large genome synthesis project, for example. And what's my vision in the 10 years? And today we heard the good news, right? The Nobel Prize goes to the structure prediction and the AlphaFold. Okay, so in this image, actually borrowed from University of Washington, and what they did is based on the small molecular, they were able to using AI to redesign the protein which are able to bind that small molecular. So what I can envision in the future for synthetic genomics is since we have a lot of sequencing data right now, and we have increased the computing powers, we also have the AI tools like large language models, which probably are going to allow us to predict the ancestor genome of an organism we are currently studying. And we're going to predict what's the organization of the genome structures, and allows us to design more controllable genomic functions in the future. And what's more, what we're going to do is we probably, in the next 10 years, we were able to do a lot of those, generate a lot of those synthetic organisms for various applications. For example, we're going to probably make the synthetic crops against the drought, the flood, temperature, and resistant to the parasite and the pests, for example. And we can also generate a lot of those synthetic microbials for the drug production and the natural products production, and also the environmental remedies, for example. That's another field I'm working on. So in the long run, I don't know, but there's many possibilities, right? And George Church proposed that you're going to revive the mammals, which has been extinct, 4,000 years ago. Okay, and recently, I and a few other scientists in China, we were talking about, okay, let's do something, we can still see it, but not extinct so long ago. We were looking at one of the species we call the Yangtze River dolphin. Okay, this is extinction recently, and they are still keeping tissues, and we can, you trying to revive those because this species has been totally eliminated from the world. Okay, and again, to do this project, what you need is again your sequencing technologies, because only if we have a complete telomere to telomere sequencing, we were able to high quality, sequencing structure, the data. Then we are able to, do the things as I wish. So that's what I'm gonna do. What my view on the synthetic genomics. So final slide is about our, outlook for the synthetic genomics. A synthetic organism, which will be a part of the biotechnologies to facilitate the biotechnologies, and of course we also have to consider the society needs, like the biosafety, biosecurity, and other things we have to consider. So that's all I want to tell today. Thank you.
Thank you, Junbiao. I was pushing you a little bit because I would like to have time for questions.
Okay.
We have 5 minutes. We have some questions. If we could get a microphone on the first rank. For a few seconds, I was transported to this dream world. I thought like cheaper beer, mammoth, T-bone steaks, and then you could like edit my genes so I never gain weight. So I really root for you. And now we take the first serious question. Thank you, sir.
Great, thank you very much for that fascinating talk. Raises lots of really exciting possibilities. I imagine that you have to engage with a lot of ethical issues as you go about doing your work. I was wondering how in practice you go about engaging with those issues and how it affects the work you do.
Yes, so that is a very good question, and I think an ethics issue is one of the first things we consider for every synthetic genome project. For example, for the synthetic yeast, even working as a yeast, there's no harmful. Maybe for beer, I don't know, maybe better. But we also, every time we were talking about a project, we involved the social scientists. They're also working on the field and to do a discussion. And we also have a group of scientists. We will do biocontainment, so trying to prevent the synthetic organisms slip into the wild. And the good thing is, every organism we're made in the lab, actually living, the fitness is worse than the natural ones. So that means we are not totally understand the system yet. And another thing we want to add is the thing we build is actually based on the blueprint of the natural system. And we were based on the natural genome to redesign, to change it, make some changes. And that actually, lead us to a lot of issues and give us a lot of knowledge to understand what the natural genome function is. Yeah, thank you.
We have another question in the back.
It's here, up here.
Oh, good.
So you mentioned AI, right? So, so AI, as we know, right? So it's evolving. It's still lots of things ahead of us, and I expect somehow in a year or 2 or 3, also big developments on the side of AI. So could you just maybe pinpoint a couple of things where this AI and the new AI with I don't know, reasoning capabilities, agent-style AI, which will master way bigger complexities as we are used today, right? How this could influence these projections which you were mentioning.
I see. Yeah, that's a very good question. Yes, so the AI has been applied in biology in many different fields. And one of the field just win the Nobel Prize is to predict the 3D structure. Okay. So for AI to work in biology, it requires a lot of data, validated data, right? So that's why I'm thinking about AI may be working for synthetic genomics, because we are able to sequencing a lot of those microorganisms, same species or same families, you can generate all those DNA sequences, right? As a biologist, you know that all these organisms were evolved from the same ancestor, right? So therefore, you can imagine the changes happened among those different organisms may be somehow related, right? So therefore, I'm thinking about using AI tools. One of the immediate things we can using to help us to design the synthetic genome is to derive the ancestral genome, for example. So, originally, the yeast, the budding yeast before it's, the whole genome duplication, what genome is, why some can make good beer, some can make, not as good as the others. So we can do these things. So that's, again, it requires a lot of those data to be generated at the first, which is the sequencing data for all those genome sequences. Yeah.
We have 30 seconds left on the clock, so I would appreciate a quick question and a quick answer, please.
Okay. We think a little bit about synthetic biology maybe as we do about programming, like a certain sequence gives a certain output. But with, for example, epigenetics, it makes things a bit more complicated. So could that be a challenge in your vision of, okay, we scale the number of genomes we can screen so we can do many things? And do you take that into consideration?
Yes and no. Okay. The yes is because for higher eukaryotes, the epigenetics plays a pretty big role in the regulation of the genome functions. Which I was a fan of the epigenetics because I spent a lot of time to working on histone modification and things like that. So yes, so for higher eukaryotes, you may have to consider the epigenetic regulations. But for the lower ones, most of them, for example, for yeast, there's no DNA methylation. You don't have to consider things like that. So yeah, yes and no. Yeah, thank you.
Thank you very much. Please come back for our next session in 30 minutes, the future of archaeology, and give a warm round of applause to our speaker, Junbiao.